Today we will be studying about Lac Operon. Lac Operon is an operating system in which lactose is used for the metabolism in the eukaryotic organism. This system, the Lac Operon, it comes from the word lactose, which shortly known as Lac Operon means system. This system was first of all detected or discovered by Francis Jacob and Jack Monod. They have discovered this system, and this system happens in the prokaryotic organisms, and basically here we will be learning the system in bacteria. So let us begin with, now since I said this system, actually it is a process by which the bacteria uses lactose for the metabolism of its system. So here what will happen if lactose is not present? In absence of lactose, what will happen? Here we will call lactose as an inducer of the system, because lactose induces or operates the system. Now when there is no lactose in the medium where the bacteria is growing, what will happen? Let us see. Here we have taken a segment of the DNA where P, I, O, Z, Y, A, these are the different genes. Here I gene, it code for the formation of the repressor mRNA, and this repressor mRNA, it keeps on forming in the bacteria cell throughout. The repressor mRNA, in turn, it will be producing a repressor protein which will bind at the site O, that is the operating site where the system will be beginning. In absence of the inducer lactose, the repressor protein will be binding here, and so it will then prevent the other genes for its from functioning. Here let us also know what these all genes code for. The Z gene, it code for the formation of beta galactosidase. Y gene, it code for the formation of permease enzyme, and A, it code for the formation of trans-acetylase enzyme. Now, what if the lactose is present in the growth medium? In presence of the lactose, what will happen? As soon as the repressor mRNA produces the repressor protein, the lactose, it will bind with the repressor protein and inactivate it. As a result, it cannot bind with the operating region O. And now the rest of the genes, that is Z gene, it will start produces beta galactosidase. Beta galactosidase, the function of it is to convert the lactose into glucose. Now this glucose will be broken down to produce energy that will be used up by the bacteria for the transcription process. The Y gene, it code for the permease. The permease, it helps in the entry of the lactose inside the cell across the cell membrane. On the other hand, the trans-acetylase enzyme, that is coded by the A gene, its function is to prevent the formation of any toxic substance during the process of the transcription. So when the inducer gene, it binds the repressor protein, so what will happen? The inducer lactose, it is binding with the repressor protein, and hence the repressor cannot block the pathway and the transcription process will be carried out. Now students, we should also understand, in order that the operating system, the Lac Operon, keeps on functioning, the concentration of lactose inside the cell should be minimum. when the lactose concentration inside the cell will be minimum, then only the permease will help the entry of further lactose into the cell, and so that the process can go on. If inside the cell the lactose concentration is high, what will happen? No longer the lactose will be entering into the cell. So this is all about the Lac Operon that we have studied. I hope the video must be helpful to you. At the end of the video, let me share one of the facts with you regarding the transcription process that happens in the eukaryotes. In eukaryotes, RNA polymerase II elongates 1000 to 2000 nucleotides beyond the end of the gene being transcribed. I repeat, the fact in eukaryotes, RNA polymerase II elongates 1000 to 2000 nucleotides beyond the end of the gene being transcribed. Thank you all.